Our first speaker today is Alia dos Santos. Alia did her undergrad at the University of Lisbon in Portugal, during which time she was awarded an Erasmus studentship. So via this award, she was able to work with Professor Kurt Rickemer at Imperial College in London, and she published a paper during that brief stint there. Thereafter, Alia worked briefly as a tech with Professor Paula Booth before joining the lab of Dr. Christopher Toseland, where she is currently a PhD student. Now, while Alia will be talking today about her most recent work on DNA damage, she has already published a number of papers from her PhD. And the great news is Alia will be defending next year and is currently looking for postdocs. So for those of you recruiting, this might be a great intro into some of Alia's work. So without further ado, thank you for presenting in this series, Alia. Take it away.
Thanks very much for the introduction. It was very nice. Yeah, my name is Alia, and the presentation is and I work in the Prison Lab as a PhD student at the University of Sheffield. Now, the Prison Lab is interested in a variety of nuclear events, from transcription, DNA repair, genome organization. But what we're also interested in, and I'll be talking a little bit about today, is about nuclear mechanics and DNA damage and the relationship between these two things, especially when it comes to cancer and cancer development. So the tissue, or the field of tissue and cell mechanics, has grown quite a lot in the last few years, as it becomes more and more important to understand how cells can behave differently, physically and mechanically, depending on cancer development stages, aggressiveness of cancer, metastatic behavior, but also in terms of therapeutics. So there's a large evidence that when we use chemotherapy drugs, tissue stiffness and cell stiffness overall change. And this really spiked our interest, because whilst a lot of these drugs have mechanisms that are nuclear, there's very little understanding on how they specifically affect the nuclear mechanics, its nuclear sensitivity, mechanical sensitivity, and conductivity. So as I mentioned, drugs like cisplatin have a nuclear mechanism, and they do this by causing DNA damage. So for example, cisplatin goes into the nucleus, creates adducts and crosslinks to the double strand helix, and this causes damage, which can be further aggravated through replication fork collapse, which can then lead to the very lethal double strand break. Now, most of you in this community will be quite well aware of this, but the ATM kinase is one of the first offenders to these double-strand lesions, and one of its functions is to phosphorylate methane H2AX, causing this or creating this gamma H2AX. I'll be referring back to this gamma H2AX throughout my talk, as we use it as a marker for the extent of damage in the cell. But really, what this does is create a hub for the recruitment of repair factors that congregate around the lesion, and if successful, can restore the genetic information that was lost via the double-strand break. So we're really interested in how DNA damage, specifically in our case cisplatin, can alter nuclear mechanics. And for this, I use atomic force microscopy. So AFM is a very powerful technique, whether we are looking at imaging with very high resolution, but also to measure the stiffness of a material. So this comes in the form of Young's modulus, which is a measurement of how much deformation we are inflicting on our material based upon a certain amount of stress. And in our setup, what we have is we have a cantilever above our cell. We choose a point directly central above the nucleus, and by pressing down, we can then get information that causes stiffness or changes in mechanics on our different cells. So we tried this with our cisplatin treatment. So we used cisplatin for hours, cisplatin 24 hours, and here we have our gamma H2AX showing a large extent of DNA damage. And what we saw initially was that between the non-treated and the cisplatin 4 hours, we didn't see very significant differences, but we can see a clear reduction in stiffness. Again, this is us looking at the central region above the nucleus. There's a reduction in stiffness of our cells or our nucleus after cisplatin 24 hours. Now, from the beginning, we were very aware that there was a limitation with our type of measurement, and this has to do with the complex cytoskeleton that is located above, below the nucleus, but also that is connected to the nucleus and exerts large amounts of force and tension in the nucleus. So what we're measuring here is really a combination of our nuclear mechanics and our cytoskeletal index. So to circumvent this problem, I used a variety of cytoskeletal drugs, such as loxatin and tranquilin B, also isolated nuclei. But again, the problem with this is that we are removing the nucleus from this physiological condition or maybe affecting other pathways that we don't want to. So in the end, and from now on, when I refer to it, I refer to microscopy data, what I will be talking about is initially it is. So here we give enough time for the cells to adhere to our surface, but not enough time that it forms a fully complex cytoskeleton. The nucleus occupies the majority of the volume of the cell and is also the mechanical limiting factor. And there's really not much difference between the mechanics of isolated nuclei and what we can measure in initially it is. So initially it itself, when we repeat this experiment with this cisplatin, whilst before we couldn't see a difference at 4 hours, we can now clearly see a softening of the nucleus. So there's clearly something occurring that is reducing nucleus thickness, and this is more pronounced after 24 hours. And this is really an intrinsic property that is being changed in the nucleus as this also is maintained when we do the measurements in isolated nuclei. So the question we asked next was this: is this a direct consequence of the damage itself of the double-strand break itself, or does this have something to do with DNA damage sensing, with signaling responses? And so we went back to this ATM kinase, and we decided to understand what would happen if we disrupted this signaling pathway. So we used an ATM inhibitor. So this no longer is occurring. And what we see is very interesting. So now we no longer see a reduction in stiffness if we treat with ATM inhibitor and cisplatin. So the cells look like mechanically behave as non-treated. So this is very interesting because it really seems like this is a consequence of the actual DNA damage sensing mechanism. So the next question was, what is causing this nucleus softening? So here we have two different hypotheses that we could follow, and this relates to what are the two major contributors to nuclear mechanics. And the major contributors are the lamina and the chromatin. So we went to look to the lamina and the nuclear envelope to see if there were changes in laminar thickness or envelope integrity. And we couldn't really see a difference between non-treated and cisplatin treatments in terms of thickness. And when we look at electron microscopy, we could also not see any lack of integrity, any rupture of the actual envelope. So this didn't seem like was the cause of our reduction in nucleus thickness. Now, when we look at chromatin, we saw a different story. So here we use electron microscopy, non-treated cisplatin, and we also use tryptophan A as an extreme comparison of chromatin decondensation. And what we can see is that our cisplatin and TSA treated populations have much higher levels of decondensed chromatin compared to our non-treated. And if we use ESM, we also see that TSA treated nuclei also have a very similar mechanical behavior compared to the cisplatin. We also used super resolution to look at chromatin and chromatin structure, and we saw something that agreed very well with our EM data, which is that with cisplatin and PSA, we have a reduction in the more condensed chromatin. So the next question, why is this chromatin decondensation important following DNA damage? Is it something that the cell requires? And one of the hypotheses was that chromatin decondensation would lead to higher diffusion and higher accessibility for the test. And we wanted to test this, and we wanted to test this using a 3D system so we could have a global picture of the nucleus. And for that, we traveled to Janelia to use a multifocal microscope, which allows us to do a single molecule tracking across 9 simultaneous Z planes. So it gives us a depth of 4 microns in Z and then 20 by 20 in X and Y. And we use the SnapTag as a fluorescent reporter to measure molecular diffusion in the nucleus in 3D. So what we have is a sort of reconstructed 3D of all the molecules that it can detect software, and then we can track these molecules and we can assign them with location and also speed and movement. So what this reporter gave us was that PSA and cisplatin actually have similar diffusion in the nucleus, and this is much higher than the non-treated cell population. So molecules, according to these molecules, can move faster, and potentially this would mean higher accessibility, even for chromatin movement that needs to occur following DNA damage, which could lead to more efficiency. Now, one other thing that we started thinking about at this point was could it be that a softer nucleus could also influence damage and how the degree of damage that the cell suffers itself? And the reason for this is that in the literature there have been lately some reports of nucleus softening having some sort of protection against damage. So we looked at this by inducing the reduction of tension in the nucleus. So by removing the cytoskeletal forces here by using blebistatin before the drug treatment with cisplatin, we remove the cytoskeletal tension force and then test how that affects damage. And here I used high content screening, and what I saw is that in cells that have reduced tension forces on the nucleus or more relaxed nucleus actually have a reduction in damage. So there seems to be a certain degree of protection in these cells compared to just cisplatin treated cells. Now, again, we wanted to do this in a way that we didn't have to use drugs. So we confirmed this using different thicknesses. So we used polyacrylamide gel as surfaces for our cells. And they correlate very well with tension on the nucleus. So gel will have higher tension on the nucleus. And what agrees very well is that we also get higher degree of damage on a cytoplasm. So there really seems to be a correlation here about how much the nucleus is under force and degree of damage that is inflicted. And this is very interesting. We are still not entirely sure what is happening. It could be a variety of factors, including replication, considering cisplatin requires replication for this type of damage. But it's very interesting that we are looking at tissue mechanics and the relationship. And with that, I'd like to thank the lab. There's less of us now than in this picture, but also Florian. I traveled quite a few times to the University of Göttingen to do my FM measurement. Everybody at the AIC at Janelia helped with the NFM and also people at Research Complex. And thank you very much for listening.
Thank you, Alia. That was wonderful. Wow, we already have 2 raised hands. So I'll remind people there's multiple ways to ask questions. You can either raise your hand and we'll give you the chance to speak, or you can type your question in the Q&A box. So I'll start with Pallabi Mustafi, who's raised her hand.
Hello? Yeah. Can you hear me?
Mhm.
Yeah. So my question was, since cisplatin is used in cancer, so in terms of metastasis, do you think this, you know, this change in the nuclear softening or in the cell mechanics, uh, is that, how is that going to also contribute in terms of uh, like when the drug should be given, or is it going to be effective depending on how the cell mechanics changes during a process like metastasis? What is your
Yeah, so
take?
there are difficulties that nuclear stiffness and tissue stiffness in general can affect migration and speed of migration. So that could definitely be something. But also, I think just be careful because depending on which type of cell, migration and what benefits and what doesn't benefit might vary. So it will depend on the type of cell, what happens.
Okay. Thank you.
All right, so I'll ask you a question from the Q&A from Matthew Levine, who says, when you relax chromatin, could you be affecting gamma H2AX condensates that may form upon DNA repair instead of actually preventing damage?
Well, yes, that is a possibility that we could be affecting the signaling rather than yeah.
Okay, all right. Genevieve Forel has her hand raised, so I'm gonna let you talk.
Hello, hi. Um, Actually, it's along the lines of the last questions, because I'm not sure I quite I heard you well, actually, and it was not quite clear to me in your last slides how you were measuring DNA damage, whether you were using gamma H2AX or some other parameter like tunnel, or really to measure DNA damage and not the DDR.
No, so here we use DNA damage as a marker. Right? We use DNA damage as a marker. So we use gamma H2AX, both in the high content and on the polyacrylamide gel. Yeah, so we're measuring a DDR, our output.
Okay, so it decreased gamma H2AX. You can't really say it decreased DNA damage, right?
Yeah.
The truth. Okay, thank you.
All right, so we have a couple more questions in the Q&A. Nicholas Sapp says, nice presentation. And he asks, did you test if cancer cells are softer than non-cancer cells?
No, I didn't test that directly. We did look very preliminary at other types of cells. But I think there's a lot of variety even within the cancer cells themselves. Some are stiffer than others. So these are HeLa cells, so they are cancer cells. But yeah.
I see. Okay. All right, the next question is from Jason Plan, who says, hi, very nice talk. I was wondering how long did the nuclear softening or chromatin decondensation last after DNA damage? Is it only until DNA repair has been completed? And do you think that non-double-stranded break damage from non-double-stranded breaks would also lead to this relaxation phenomenon?
So we didn't try and do the recovery of the cell, so I don't really have a feel for how long it takes to recover. Also because some of the treatments are quite long, so the cells are quite damaged by then. We tried other types of damage. We didn't look at chromatin decondensation in the same sense. So I'm not sure, but I think that there are some other groups that do some very interesting work on different types of damage and protein remodeling that can occur after that.
Excellent. Thank you, Alia.